name is Abhishek and welcome back to MBBS Treasure channel. In my previous videos, I have already introduced you to allergy or hypersensitivity, and I have also explained you about the classification of hypersensitivity reaction. So in today's video, we are going to learn the first type of hypersensitivity, that is about anaphylactic type of hypersensitivity reaction. Okay, so this video is very important because it will help you to build your concepts about hypersensitive reaction, and also it will help you in croc. And this very topic is also asked in the module exams by our teachers. So don't worry about the topic of today's, and please watch till the end because this video is going to be a bit longer, so please watch till the end. You will get a very good concept about the hypersensitivity type 1 reaction. Okay, so without wasting time, let's begin our today's topic. So let's see what are present in this board, and it's looking very complex, but don't worry, it's very easy. And if you will understand the topic according or diagrammatically, if you will understand or you will feel the topic, then only it will persist in your brain for a long period of time. So I made these diagrams or making it a bit complex to look, but I say you it's very easy. Let's begin our today's topic. So this hypersensitivity reaction or the allergy reactions occurs when there will be some kind of allergen in our environment, and this reaction is known as anaphylactic reaction, otherwise known as reagin reaction or ATOPY reaction, or it is also called IgE, that is antibody E mediated immune reaction or hypersensitivity. It is the most quickest form of hypersensitivity reaction, so it is categorized under immediate type of hypersensitivity reaction. This anaphylactic is otherwise known as this IgE mediated, and its other name is reagin. Why? Because the IgE in Latin term is known as reagin. So that is the reason why this anaphylactic hypersensitivity is otherwise known as reagin type of hypersensitivity. Next, this total hypersensitivity or the different types of hypersensitivity occurs in 3 pathogenesis stages. And what are these 3 pathogenesis stages? It means it will be going under certain circumstances, and then only our body will react to the pathogens or allergens. What are these? First one is there will be immunoreaction, second one is pathochemical changes, and third one is pathophysiological changes. So these are the 3 stages, or this whole hypersensitivity reaction will occur in 3 stages. First one immunoreaction, second one is pathochemical reaction, and third one is pathophysiological changes. So first of all, these reactions I have made in diagrammatical presentation so that it will be easier for you to understand. So first of all, what will happen when there will be first time exposure to the allergen, when there will be first time exposure of allergen to our body, what will happen? This is the allergen that I have made. When it will first be penetrating our body, let's say it penetrates through our nasal passages, and here are the mucosal membrane of the nasal passage, and there lies a specific cell. What is that cell? Yes, it is antigen presenting cells. They lies in the lining of the mucosal membrane, and what are their function? They will trap the allergen coming through the nasal passage. And what will be their function? They will modify the allergen coming into our nasal passage, and they will provide to certain cells like the T lymphocytes and the B lymphocytes. Their function is to present the allergen. So they are known as antigen presenting cells. Okay, so these are the antigens that will be presented by this antigen presenting cells. After the antigen presenting cells will catch the antigen, what will happen? It will also undergo certain stages inside the cell. What are the changes that it will take? Like, first, when the allergen or the antigen is coming, then it will bind to this APC. Here is the APC, and this antigen presenting cells will try to phagocyte the allergen or antigen through passing through its membrane. Then what happens? These are some small cells which you can see. These are known as MHC type 2, major histocompatibility cells type 2. So the next question should come to your mind, why this type 2 and what is that MHC? MHC means it's a major histocompatibility. These are the proteins that are present inside the vertebrate DNA, deoxyribonucleic acid. And why they are present? They are specifically present for this kind of immune cell reactions. They need to present the cell surface proteins to the next surface of the macrophage or the antigen presenting cells. Okay, so these are the functions of this MHC, that is otherwise known as major histocompatibility complex. Okay, so these are present in more types, like they are present in type 1, type 2, type 3, but mostly this MHC type 2 will try to bind with the coming allergen and it will form a structure that is known as determinant. Okay, here there will be a complex structure formed after the binding of this MHC complex and the allergen, and that is known as the antigen will be processed by this APC. It will be converting to a determinant form. Determinant form means it's a form that can be recognized by the upcoming cells. For the further process of these complex reactions, a determinant or a structure, complex structure will be formed. Okay, now what will happen next? This determinant along with this MHC compound will try to get to the next surface of the plasma membrane of this antigen presenting cell. This antigen presenting cells is nothing but the macrophage. As I have said that its function is to analyze and process and present the allergen to the next upcoming cells. So next what we will see, this whole determinant structure will be expressed in the next surface of this antigen presenting cells or the macrophage. So this process is known as expression. This whole process, like the allergen coming and getting converted into the determinant form and then expressing in the next surface of the antigen presenting cells, is known as expression. Then what will happen? As we know, after the process of expression, the macrophage transfers the message to Th2. It means the T helper 2 cell. It means that there are present like the CD4 receptors on the surface of T lymphocytes, and these receptors which are present on the surface of T lymphocytes will recognize, yes, the antigen presenting cell is presenting something and I need to take that information. And why the Th2 cells? You should be thinking that why I have written these two, because Th1 means it is a suppressor of T lymphocytes for production of antibody. Okay, so if Th1 will be activated after the antigen presenting cells, so this whole process will not take place. Rather, the antigen presenting cells will try to present the determinant form of antigen to the next cell, that is Th2 cell or T helper 2 cell, where there lies this CD4 receptors for accepting the messages. Okay, so this appearance of this complex structure is known as expression and then passing the messages or informations to T helper cell. Okay, so next what will happen, this whole structure like the T lymphocytes or the T helper cell will be also activating certain cell. After that, what is that cell? As we read that it will activate B cell for the process of formation of antibodies. How it activates? Yes, when the T cell activated, okay, then next what will this whole complex will be reversed and it will be taken the same informations to the next cell, that is B lymphocytes. Also the T lymphocytes will produce certain kind of chemicals from them, like known as cytokines. All together they will try to activate this B cell, like the B cell will get activated through this message transfer along with this determinant form. Okay, next what will happen, this antibody as the B cell will get activated, then what will happen, there will be formation of lymphoblast and the plasma cells. It means the B cell activated and it will get activated after this whole process and will form the lymphoblast cells. What are lymphoblast? These are the stem cells, or these acts like stem cells for producing or for proliferating more number of B cells. Okay, so as the activation of B cells takes, it will proliferate, more number of B cells will be forming from the lymphoblastic cells, as you see the very name, the blast cells. Okay, then process takes place is proliferation. After that, what will take place? The blast cell transformations. From the very lymphoblastic stage, it will get proliferate and differentiate to a certain specific cell that we call as plasma cells or the plasmacyte cells. These are the cells responsible for producing the antibodies and they are known as IgE producing cell of B lymphocytes. So these plasma cells or the plasmacyte cells are the actively forming antibody or producing the antibodies. Okay, so what will happen, this here in this process there will be transfer of message, it will get proliferated and differentiated through the blast cell transformation process. Okay, so these plasma cells will be producing the antibody. Okay. Next what will happen, this is the plasma cell. I am giving more stress to this IgE. Why? Because you should remember that this anaphylactic or the immediate type of hypersensitivity type 1 is occurring only because of IgE, antibody E mediated. That is the reason why it is known as reagin type. Okay, so it should be in your mind that it is mediated by IgE. So next what will happen, this IgE will be released and this antibody will be released. Where it will be released? As it is present, as the whole process is taking after the cell layer, after the mucous membrane. So the nearby tissues will be flooded with these antibodies. Also it will be moving to certain vessels, small blood circulatory vessels. So this antigens, these antibodies, after the production, it will be trapped or it will be highly having affinity towards a certain specific cell that is present around the tissue, that is known as mast cell, and it will be having high affinity. The antibody will be having high affinity towards some cells that are present inside the vessels. What are they? They are basophils. Okay, so they are basophils. Here the antibody will try to say the body that yes, something is wrong occurring, that is the reason why I am being produced, and it will be traveling throughout the whole circulatory mechanisms of our body and traveling or giving the message to whole body that something has occurred, some changes or some pathological condition is trying to develop in our body. That is the reason why some antibodies will try to go to the vessels and to the surrounding tissues that are present, and if it is present or surrounding tissues, it will go to mast cell. This whole process where there is increasing in the production of IgE or antibody E. It occurs specifically in condition of allergic reactions, when there will be some types of allergens entering in our body, and specifically when there will be parasitic infestations. So this process will occur, whole process will occur. Next, this total production of IgE and getting attached to certain specific cells is known as sensitization. So next question comes is that why I am stressing the word sensitization. What do you mean by this sensitization and what are its effects? Yes, this very process is very much essential, because until and unless this sensitization of the cells like mast cell or the basophil cells present inside the vessels or tissues will not occur, the further process will never occur. And if this sensitization is not going to occur, then our body is never going to get protect from these kinds of allergen or the antigens. So this very process, the sensitization is required for the production of, or for more increase in the production of antibodies like IgE in response to the allergens. Okay, so this process is important, and this process occurs when there will be increasing in the IgE and it will get bind to the mast cells. Next comes the sensitiveness to allergen. As this sensitization occurs, so the sensitiveness to the allergen is divided according to two types. It can be active and it can be passive. Listen, active means if our own body is actively producing the antibodies against the allergen or antigen, then we will call it as active sensitiveness to allergen. And passive sensitiveness, if our body is not able to perform these cascades of reactions, if our body is not able to recognize what kind of allergen is coming, so this process will not occur. This transfer of messages through different kinds of cells will not occur. There will be no formation of the plasma cells or the plasmacytes. So there will be no formation of these IgE cells. Okay, so this is very much essential for those individuals that we need to give the ready-made Ig. Means we need to give them the pre-formed antibodies to their bodies for fighting these reactions, to fight against the allergen. So in that case, when we give the ready-made antibodies or when it is introduced to the individuals whose body cannot process these all types of cascade reactions, then we will call it as passive way of sensitiveness to allergen. It means that we are helping the cells of our body, or we are helping the person's or individual's body to sensitize. And this sensitization is very essential, because this IgE means this antibody can only sustain, or it can only survive for 2.5 days, not more than that. But if the allergens is coming for more than one time exposure, and it is coming continuously and it is getting exposed, our body is getting exposed for more than once, then what will happen? What will our body do? This sensitization will help. After the sensitization of the mast cells will produce certain kind of chemical mediators or the chemical reactors in response to the whole process. And for what? For helping. It means they need to resist. It will help the body to resist the total effect of allergen by producing certain kind of mediators. So in next process we are going to read those, what are the mediators, how it is produced. Okay, we now learned this, what do you mean by sensitiveness to allergen. Next, the IgE persists for 2.5 days, as I have said. But why I have written but? Because the sensitization whole process remains for 6 weeks. Okay, so we need our body to interact or to react to certain kind of situations for more than 2.5 days. That is the reason why we need the sensitized mast cells or the sensitized basophils. Okay, so next comes the next process, what happens when there will be increasing or when there will be increase the exposure of the allergen for more than once. If our body is getting exposed to allergens, what will happen? These are the allergens, okay, so these are the allergens and it is continuously getting exposed. So what will happen after the whole process? The extra allergen will try to bind with the mast cells along with the antibodies. Okay, so these are the antibodies, IgE I have represented, and the mast cells have a specific receptor, that is the Fc receptor. We are going to talk about that right now. This is the Fc receptor. These are the Fc receptors that are present on the membrane surface of the mast cells or the basophil cells. What are their functions? They have specific receptivity towards the antibody IgE. Okay, what they have? They have four structural compartment units like alpha 1, alpha 2, beta 1 and beta 2. Okay, so these are the four structural units that when they will combine, then they will form these kinds of receptors. And what are why they are present? They are present for receiving the specific IgE antibody. Okay, you can see the structure over here. It has two alpha units, alpha 1 and alpha 2, and two beta units, beta 1 and beta 2. Okay, so the next question comes here is that if it requires or if it receives the IgE, then it should have certain kind of affinity. And what is that affinity? Yes, the Fc receptor has two kinds of affinity. First one is Ig epsilon R1 and Ig epsilon R2. Why I have said, or why is it epsilon? Okay, so there are two kinds of receptors like R1 and R2 according to Fc receptivity, where the Ig means the antibody has very high affinity towards the R1 receptor, and towards the R2 receptor it has low affinity. Let's say, let me draw one example. This is the antibody. Okay, so it has two chains. One is heavy chain and the second one are the light chains. The purple color is the light chain and the red one is the heavy chain. So it has two receptivity, like the first one is the R1 and second one is R2. Let's say, what do you mean by this? The heavy chain is represented as epsilon, okay, and the light chains are represented as lambda or kappa. So there, as we know the structure of antibody, it contains two light chains and two heavy chains, and they have different kind of representations. So in Latin form, these heavy chains are represented as epsilon, and according to their positions they have two receptivity, R1 and R2. So R1 receptivity with this Fc has high affinity and R2 has low affinity. These are the extra things that you should know about the concepts. Okay, so next, what will happen when there will be extra or continuous exposure of allergen? What will happen? The allergen will come and bind to the corresponding antibody that are already bound with the Fc receptor of the whole mast cell. Next, what will happen after the binding of this allergen or antigen to the antibody? It is an antigen antibody complex will be formed, and you can see a special structure. It is first antibody and it is second antibody, but the allergen is placed in between them. So what is it doing? It's forming a cross bridge, and this whole process is known as cross bridge formed between the two antibody. This process or this cross bridging will help, or it will stimulate the next processes that will be occurring in the mast cells. So what are the next processes that will be occurring in the mast cells? Yes, after the binding or after the bridging that will be formed in between, there will be active influx of calcium. It is an active process. You should remember this. It is an active process where there will be influx of calcium. Due to influx of calcium, what will happen? the there will be certain kind of chemical reactions that will be occurring inside the mast cells. What will happen? It will activate the phospholipase A2 enzyme, and then it will activate this arachidonic acid metabolism. This is a special kind of cascades of reactions that occurs in most of the inflammatory reactions that we are going to read in next chapter, that is inflammation. So I will just say a little bit about this arachidonic acid metabolism. This calcium influx will stimulate this acid or arachidonic acid metabolism processes. It occurs through cascades of processes through which at the end there will be formation of these chemical mediators, and these are nothing but the mediators of inflammations that will cause or that will show the manifestation in our body due to the inflammations. Okay, so they are called as mediators of inflammations. Next, how these mediators are formed? When the mediators are formed, they will gather and there will be a formation of a structure like a vacuoles, and that vacuoles are nothing but this granular in form, and these granules will try to diffuse to the substance or diffuse to the surface of the mast cells, and then they will release around the mast cells of the mediators. Then they will release those mediators that are present inside the vesicles or the granules. Okay, so next, this process is known as degranulations. So when the antigens or the allergen will come and bind to the bridging surfaces of the both the antibodies, it will be forming the cross bridge, and that whole process will stimulate the production of these mediators, and these mediators will bind to the next surface and will get released. So this is the whole functions of the mast cells. Mast cells are the most important cells in the whole process, that are the leading cause of production of these mediators, because these mediators are the only ones that will cause the effects in our body. These mediators will cause certain kinds of pathophysiological changes in our body that we can see as clinical manifestation, or it can be clinical symptoms. It could be redness, itchiness, inflammations, anything. So these are the mediators are the reason, and that we will be reading after this slide. Okay, so next, these mediators are released, and this whole process means when there will be continuous exposure to the release of the mediators, this is the stage 2 of the pathochemical changes. As the chemicals are secreted, it is known as pathochemical changes. First one was the immunoreaction. How the immune cells like T cells, B cells are reacting and producing the antibodies. Then the sensitization, and then after the sensitization, how the antibodies or the mediators are produced from the mast cells, leading to the stage 2 of the pathochemical changes. Next stage comes is the stage 3 or pathophysiological changes. What happens there? After the release of the mediators are classified according to two types. First one is the early phase mediators. That means when the process of when it will stimulate the production of mediators, in few seconds these mediators are going to produce. So they are classified under early phase mediators. And late phase mediators means after 5 to 10 minutes the mast cells will produce these kinds of mediators, and each of the mediators have their different functions. We are going to read them. So let's name them. What are the mediators that are present in early phase, and what are the mediators present in late phase? In early phase, the mediators like histamine, prostaglandin, PGE2 specifically, and leukotrienes and factor activators of thrombocyte, otherwise known as FAT, and some kinds of polypeptides. SRSA means slowly reactive substance of allergy. Okay, and this polypeptides, the whole things will be coming under this early phase. And next comes the second is the late phase. It means when the mediators will be produced after 5 to 10 minutes. What are these examples? It is like histaminase, prostaglandin E1 and E2 both. The prostaglandin E2 that will be produced in few seconds will come under the early phase, but after some time the mast cells will again produce the prostaglandin E1 and E2 that will be coming under the late phase. And next come the aryl sulfatase, phospholipase B and D, polysaccharides, mediators, basic proteins, eosinophils. These are the mediators will be having specific reactions in our body that I am going to tell you in my next slide. Okay, so these changes that will come under the stage 3 pathophysiological changes. Okay, so in this total we learned the stage 1 immunoreactions, how the immune cells are reacting. Then comes the sensitization along with the expression, along with the expression and the blast cell transformation. The antibodies are produced, and after the antibodies, this sensitization of the mast cells and basophil cells takes place. After sensitization, the specific process, the degranulations takes place. Degranulations through which the granules will break and it will produce more number of mediators. Okay. So it will be producing more number of mediators. And next, the mediators will be classified according to early phase and late phase. In my next slide, we will be reading about these mediators, specific actions, and what are the stage 3 pathophysiological manifestations. There are two: local manifestations and clinical manifestations. Okay.